In this session, we'll be talking about a very important means of reproduction of cells. This means is called the cell cycle, and it is used in order to make more cells identical to the original parent cell. It's going to be a little bit different than a cycle that we'll talk about a little bit later on in a future session. In the cell cycle, we're producing cells either to make additional cells, like the original parent, or we may be making cells to repair damaged cells. And in some organisms, not all organisms, these organisms can regenerate lost body parts using this method of cell reproduction. As I said, this method is called the cell cycle. And this cell cycle involves several different steps. The first step is referred to as interphase. Interphase, for many, many years, was thought to be a resting phase. No longer do we know that that's true, because we know that it is, in fact, a stage in which many things are prepared for future steps in this cycle. So we could more correctly consider this interphase as a preparatory phase for what comes after. There are three subphases that occur during interphase. There is a G1 or gap 1 subphase. There is an S subphase, and there is a G2 or gap 2 subphase. The G phases produce things primarily that will be needed for growth of the cell. The S subphase is often referred to as a synthesis subphase, and this is where we make a copy of the DNA. Now, why do we need to copy the DNA? Well, if we're going to begin with a diploid parent cell, we designate that as the 2N number for this organism, diploid, and if it's a human, that will be 46 chromosomes. And we're going to make new cells that contain 46 chromosomes. Then at some point in time, the DNA must be replicated so that there will be enough information material to share between two identical daughter cells. So somewhere between here and here, there must be an S subphase so that we have enough information material. Once a cell completes interphase, and this is the longest stage of the cell cycle, once everything is made for the future stages that occur here, then the cell will move into prophase. There are a couple of things, if we're looking at slides, that we can look for in prophase. Two of those things are we can see the nuclear envelope begin to disintegrate. I spelled that wrong. Disintegrate. Let's start again here. And we can see the chromosomes shorten, thicken, and they become a lot more distinct during this stage here. So it is a significantly different thing that we see here in prophase as opposed to interphase. Because in interphase we see a distinct nuclear envelope, and we see the DNA in chromatin form, or thread-like form. Once a cell completes prophase, it will move into metaphase. Classically, we recognize metaphase as the chromosomes align at a midplate. Chromatids align at a midplate. Then in anaphase, we see those chromatids separate, and then finally in the last stage here of the cell cycle, telophase or telophase, we see the cell actually split in two as cytokinesis occurs. Now everything that happens from interphase down through anaphase accomplishes what we call karyokinesis. We divide the nuclear contents, basically the chromosomes here in karyokinesis. But in this last stage of the cell cycle, we're going to divide the cytoplasm, and we call that cytokinesis. And cytokinesis is different in plant and animal cells. Plants accomplish cytokinesis with the formation of a cell plate. So if we have the chromosomes at opposite ends of the cell here, a cell plate will form, beginning in the inside and moving to the outside, so that ultimately we will divide using that cell plate. We will divide so that we have two new daughter cells, exactly like the original parent cell in chromosome number and makeup. Animal cells divide differently. Animal cells form a cleavage furrow. And basically what that cleavage furrow looks like is this. We have the cellular contents at each side, at the end of anaphase, at each pole in the cell, and now the cell begins to pinch inward. And that cell will pinch inward, forming that cleavage furrow. It's sort of like pulling a drawstring across the cell. And the ultimate result will be, as the cleavage furrow forms, we will divide the original parent cell now into two identical daughter cells. These cells and these cells will be diploid cells, just like the original parent cell. Cell cycle is carried on by eukaryotic organisms, eukaryotic cells, to produce more cells just like the original parent.